Okay, I'm going to try to make a video here that will go over all the information that you need to know for the second exam. And this is going to be a one take. So I'm going to be going through the module text, briefly explaining concepts, and hopefully this will get you all the necessary information that you need to know to get an A on the exam. Like I said, this is a one take, so bear with me. I'll probably be maybe not super clear on all of the material, but hopefully most of it. All right, let's get started with Module 5 on metabolism. Okay, so metabolism just refers to the chemical processes that cells use to take in energy and release that energy. The processes that we're really focusing on is the breakdown of carbohydrates and glucose being that primary carbohydrate. The chemical formula for glucose is C6H12O6, and we combine that with oxygen in animals to release carbon dioxide, water, and energy. The opposite happens in plants during photosynthesis. We take in carbon dioxide, or rather the plants take in carbon dioxide, water, and energy from sunlight to generate glucose and oxygen. And during all of this, we have two primary forms of energy currency really, that are being generated or consumed in the form of ATP and NADPH. There's also FADH2, but that's pretty minor. So let's continue here. Okay, so two things you need to know. Anabolic means synthesis, so taking simple things and making them into complex things. So the synthesis of glucose in plants would be anabolic. The opposite is catabolic, which is what we do during cellular respiration. The breakdown of glucose would be considered catabolic. Okay, so types of energy. Well, there's really two types to really be aware of: kinetic energy and potential energy. Kinetic energy is simply energy in reference to something that is in motion. So, like a car that's going down the highway, it has kinetic energy. Anything related to potential energy would be, if you take physics, you know that anything that's up in the air has potential energy. Potential energy could also be in reference to chemical energy within the bonds of compounds like glucose, for example, or even something on a more macro sense like an actual food item would have a lot of potential energy. Gasoline, oil has a lot of potential energy stored within its bonds. And a measurement of this energy would be Gibbs free energy. The letter that we use for that is G. And an important thing to know is that whenever we convert energy from one form to another, a small amount or a large amount of that energy is lost in the form of heat. We call this entropy, and the universe is always generating more and more and more entropy, more and more disorder, because things always go from a state of harmony to disorder. Just think about your room. Over time your room gets messy, but it doesn't magically become clean again. You have to exert energy to make it clean again. So entropy is a form of disorder. So all chemical reactions result in a change in energy we call that delta G, change in energy, change in free energy. So for example, when you burn gasoline in your car, the products of that combustion have less energy than the reactants, which would be the gasoline that you burned. So when delta G is less than 0, that means the products have less free energy than the reactants. So think of the gasoline. When delta G is greater than 0, the products have greater energy than their reactants. Think of this as the synthesis of glucose in plants. And the total energy change is called enthalpy, which is change in delta H. Okay, and any reaction that has a delta G of less than 0 would be exergonic because it's releasing energy in bonds. Anything with a delta G above 0 is endergonic because it takes in energy to form those more complex bonds. Another thing to be aware of is that negative delta G reactions are spontaneous, whereas endergonic reactions, which have a delta G above 0, are not spontaneous. Think again, your room doesn't spontaneously clean itself. You have to put energy in to get it clean again. Okay, equilibrium just refers to a state of total and complete entropy, and living systems are constantly in a battle with entropy. Entropy is our enemy, and we constantly have to take in energy in order to fight off this equilibrium. So when we're in a state of total, complete equilibrium and entropy, we're probably dead. The last thing in this section would be this chart. You should really know how to interpret. This first section on the left is a exergonic reaction because we end up with less free energy than we started with. And the one on the right is a endergonic reaction because we have more energy than we started with in the products of that reaction. This hump represents the activation energy that is needed to get that reaction to even start. And when stuff is in this interim phase, this humpy, whatever you want to call it, it's in a transition state. Again, exergonic reactions are spontaneous. Endergonic reactions are not spontaneous. Okay, let's talk a little bit about thermodynamics. The first law of thermodynamics essentially says that energy cannot be created or destroyed. It can only change forms. For example, light energy changing to chemical energy within a plant. Second law of thermodynamics, essentially, it says what I said earlier, that we always move toward entropy, additional disorder. Every reaction will make it so that the universe has a greater amount of entropy because that some of the energy is lost to heat, which increases the entropy of maybe not that system, but it does increase the entropy of the overall universe. So, like I said at the beginning, ATP is the energy currency of a cell. It's 3 phosphate groups attached to a ribose sugar and a nitrogen base. And these bonds between the phosphate groups are called phosphoanhydride bonds. And when we take ATP and water, we create free energy. The opposite is the case with ADP, which is the synthesis of ADP and a free inorganic phosphate plus energy will give us our ATP again. This is what happens during cellular respiration. And ATP is dephosphorylated to essentially power cell functions. One of those could be the sodium-potassium pump, where a phosphate group is transferred from ATP to the pump and gives it the energy that it needs to perform its function. I guess some key numbers to know. ATP releases 7.3 kilocalories per mole, and actually that is even greater within living cells. It's actually 14 kilocalories per mole energy released, or that's also 57 kilojoules per mole. Okay, let's talk about enzymes. Enzymes reduce the height of the humpy. They reduce the activation energy for reactions to take place. Without enzymes, reactions would take a very, very long time to take place. Enzymes are important to speed up these reactions by lowering that activation energy. Enzymes bind to substrates, or you could also say that substrates bind to enzyme active sites. And note that enzymes only work during ideal conditions. If the pH or temperature changes, they could denature and become useless. Okay, this idea of induced fit, it's think of an enzyme giving a substrate a nice hug. So the substrate comes close to the enzyme, and it kind of when it locks in, it forms around its substrate, gives it a nice little hug, and then the reaction takes place and the products are generated. So induced fit, just think of it like a hug. The enzyme is given the substrate a nice little hug. Okay, so how do you actually regulate these enzymes? You can use inhibitors. You have competitive inhibition and non-competitive inhibition, also called allosteric inhibition. So competitive inhibition is when an inhibitor essentially competes for the active site on an enzyme. And this is not as effective as a non-competitive inhibitor, because if there's a lot of substrates out there, then that competitive inhibitor is going to have a harder time slowing down the reaction. Whereas a non-competitive inhibitor doesn't try to go for the active site, it just goes for some other site called an allosteric site on the enzyme, and as soon as it binds to the enzyme, it changes the shape of the enzyme and makes it not able to bind to its substrate. Similar with allosteric activation, when an activator hits that allosteric point, it makes it so that the enzyme is able to take in the substrate. It changes the shape back to where the enzyme is able to bind to that substrate. A couple things we need for enzymes to function: helpers, essentially cofactors, which are inorganic, like iron or magnesium, and coenzymes, which are, like vitamins. Vitamins are pretty much the primary example of coenzymes. Lastly, we have feedback inhibition, where this prevents reactions from getting out of control and make sure that the cell doesn't waste energy. So one example of a feedback inhibition is when the cell has a lot of ATP, it doesn't need any more, that ATP actually inhibits the pathway that creates ATP. So it's a self-regulating system where the product actually can stop the generation of itself if there's too much of that product. And that's Module 5. Module 6 is all about cellular respiration. So let's go for it. Okay, so cells and cellular respiration, and well, it happens in photosynthesis as well. We get these reactions called redox reactions, which are oxidation and reduction reactions. Basically, all you need to know is that a oxidation reaction, it has nothing really to do with oxygen, as I thought in the beginning. No, it doesn't have to do anything with oxygen, and reduction doesn't really have to do with anything being reduced, or it doesn't make any sense. Scientists are bad at naming things. I don't know why they're so bad at naming things, but they are. But anyway, oxidation and reduction have to do with electrons. Oxidation means that electrons are being removed from something, and reduction means some electrons are being added to something. So a reducing agent is something that reduces something else. So NAD+, for example, is something that reduces something else, and it ends up being reduced to NADH. And something that oxidizes or takes electrons away from something else, that would be like NAD+. So NAD+ would be a oxidizing agent because it is the thing that is being reduced. So I'll go over that again. A reducing agent is something that reduces another thing. An oxidizing agent is something that oxidizes another thing. And reducing means it's getting electrons. Oxidizing means it's losing electrons. Already talked about ATP. I talked about dephosphorylation, which is when ATP is broken apart. Dephosphorylation releases energy stored within ATP. And okay, so substrate level phosphorylation is the primary first step, I guess, in ATP production. Substrate level phosphorylation occurs during glycolysis. This is very important to know. Substrate level phosphorylation occurs during glycolysis. So it's basically the source of ATP for a cell that if the cell doesn't have any oxygen, it can still generate some energy through substrate level phosphorylation. And when we start with a glucose, we can actually break that glucose down and get some ATP from it. About 10% of the glucose's energy can come from substrate level phosphorylation through glycolysis. But the majority of ATP production, or 90% of it anyway, comes from oxidative phosphorylation, which requires oxygen. Oxidative, it requires oxygen. And that would be chemiosmosis, where those hydrogen ions flow down their electrochemical gradient to generate ATP from ADP. So the first step in cellular respiration is glycolysis. Glycolysis occurs in the cytosol of the cell, and it does not use any oxygen, and we call that anaerobic. That means no oxygen required. So glycolysis, of course, starts with glucose. Makes sense, the name, glycolysis, glucose. And to actually get this started, we need a little bit of ATP to jumpstart this reaction. So we actually need 2 ATPs to jumpstart this reaction. And this glucose molecule, after 2 ATPs are invested, turns into fructose 1,6-biphosphate, which is very unstable and will break down into glyceraldehyde 3-phosphate. And it's important to know that we get 2 of those. We get 2 glyceraldehyde 3-phosphates for every glucose. So from now on, we have 2 glyceraldehyde 3-phosphates. So those 2 glyceraldehyde 3-phosphates are able to reduce a NAD+ to NADH, and that means we get 2 NADHs, and those are further broken down to pyruvate. And through the whole course of glyceraldehyde 3-phosphate being turned into pyruvate, we generate 1 NADH and 2 ATP for each glyceraldehyde 3-phosphate. So we actually generate 4 ATP and 2 NADH from those 2 glyceraldehyde 3-phosphates. However, we also spent 2 ATP to get this process started, so glycolysis essentially results in a net result for the cell of 2 NADHs and 2 ATPs. And remember, we are left behind with 2 pyruvates as well. Okay, so now we have these 2 pyruvates. What happens then is that pyruvate gets oxidized and a carboxyl group is removed from it, and we generate another NADH from NAD+. And through this process, pyruvate is changed into acetyl-CoA, which will go into the citric acid cycle. So in the oxidation of pyruvate, we get another 2 NADHs from our original 1 glucose, because remember, we have 2 pyruvates, so this oxidation happens twice. And this is also where we get our first carbon dioxide leaving the cell. And we actually get 2 carbon dioxides because we have 2 pyruvates. So now we're left with 2 acetyl-CoAs. And those 2 acetyl-CoAs go into the citric acid cycle. So those 2 acetyl-CoAs get combined with oxaloacetate to form citrate. And then through a series of redox reactions, we generate 1, 2, 3 more NADHs, 1 more ATP/GTP, and 1 FADH2. And remember, we have 2 acetyl-CoAs, so we actually generate 6 NADH, 2 FADH2, and 2 ATPs in the Krebs cycle, citric acid cycle, whatever you want to call it. So now we have a bunch of NADH, and that NADH will be further used in the electron transport chain. Now, it's important to know that this citric acid cycle, again, does not produce much ATP. It actually produces even less ATP than glycolysis, and it also does not directly use any oxygen. So where do we actually use the oxygen, or why do we need oxygen? Well, it's because of oxidative phosphorylation. So this occurs in the mitochondrial inner membrane and the mitochondrial matrix. So it occurs in the mitochondria, not in the cytosol. Same thing with the Krebs cycle, citric acid cycle, occurs in the mitochondria, does not occur in the cytosol. The only thing that occurs in the cytosol is glycolysis. So in the electron transport chain, we finally oxidize the NADHs, and the energy in those NADHs are used to pump hydrogen ions, or just protons, whatever you want to call them. It pumps hydrogen into the inner membrane space, and it kind of functions like a relay race where the electrons are getting transferred from one complex to another with these electron carriers, which function inside the inner mitochondrial membrane. The one you want to know the name of that moves these electrons between these complexes, the one you want to know is called ubiquinon, whatever, I don't know how to pronounce it. Probably should know the name of this one. And the whole goal of this system is to pump these hydrogen ions across into the inner membrane space. So in the inner membrane space, we're going to have a lower pH because we have more hydrogen ions. And what do we do with that concentration gradient that we just created? Well, we use that concentration gradient to actually generate ATP from ADP. Those hydrogen ions are going to flow back down their concentration gradient from the inner membrane space back into the mitochondrial matrix to phosphorylate ADP to make ATP. And this is where the majority of the ATP comes from chemiosmosis, and finally using all those NADHs to pump those hydrogen ions and eventually use those hydrogen ions, potential energy stored in that concentration gradient, to phosphorylate ADP to make ATP. And we make about 32-ish ATPs from every glucose. In the case that we don't have oxygen, we can still make energy in a process called fermentation. The reason we need to do that is because when we actually generate those ATPs from glucose, we're constantly generating these NADHs, which are not being used. And so we need to get back our NAD plus to allow the glycolysis cycle to continue. And the way we do that is through fermentation, where we actually reduce NADH in a different way. Instead of the electron transport chain, we reduce it through fermentation, where that pyruvate is converted into lactate, and then we get our NAD plus back, and the glycolysis cycle can continue. This is a very low energy producing process, which is why we can't maintain it forever. We generate a lot of lactic acid in our muscles, and that's how we feel tired, or that's why we feel tired, rather. But know that it is possible to generate ATP without oxygen through fermentation, and know that fermentation allows us to reduce NADH back to NAD plus and form lactate from pyruvate. All right, now let's talk about photosynthesis. All right, so photosynthesis basically means getting energy from sunlight, and we call those photoautotrophs, or just autotrophs. We call things that eat these photoautotrophs or eat other things, we call those heterotrophs. Chemotrophs basically means we get energy from inorganic compounds. These would kind of be like bacteria at the bottom of the ocean where there's no light. They would be like chemoautotrophs. So here's the chemical equation for photosynthesis. Probably pretty important to know. Carbon dioxide plus water plus energy from sunlight gives us sugar in the form of glucose and oxygen. Okay, so where does photosynthesis take place? It takes place within the chloroplasts of plants. So they take in carbon dioxide through openings in the bottom of their leaves called stomata. And inside the chloroplast, we have these discs called thylakoids. And those thylakoids have a lumen inside of them. And they also have chlorophyll inside of them, the thylakoids do. A lot of thylakoids is called a granum, and the fluid inside a chloroplast is called the stroma. So there are two different, sets of reactions you need to be aware of for photosynthesis, the light-dependent ones and the Calvin cycle, or the dark cycle. Probably best to just call it the Calvin cycle. So in the light-dependent reactions, this is what we actually use water and CO2. I'm sorry, just water. We just use water in the light reactions. And we use sunlight, and we generate ATP and NADH. And then that goes to the Calvin cycle to fixate carbon dioxide and generate our glyceraldehyde 3-phosphates, which are used to make glucose. And then those oxidized ATP and NADPH go back into the thylakoids to be reduced again, and the cycle continues and continues. Okay, so before going into photosynthesis, we'll talk a little bit about light energy. Light energy is transmitted through photons. Photons have a wave form. They're like a wave, like a sound wave almost, but light wave. They're light waves. And there's a big old spectrum of these light waves. And the faster these light waves oscillate, the higher their frequency of their oscillation. If you can see my mouse, that means that the higher energy they are, the faster their frequency. And it goes from gamma rays being the most powerful to X-rays to ultraviolet to visible light to infrared to radio waves. So inside plants, these chlorophyll actually absorb this light energy. Usually the visible light is what they absorb. And the reason why we see plants as green is because they're absorbing light that isn't green. They're actually reflecting the green light. They're absorbing light with wavelengths that are not green. So know that plants that are green reflect green light. So there are actually two forms of chlorophyll, chlorophyll A and chlorophyll B. Just know that they absorb different wavelengths of light. That's pretty much all you need to know. Carotenoids absorb other forms of light, like they can be orange, like orange of an orange peel is an example that's given. They absorb, different wavelengths of light as well. And the type of wavelength that a pigment absorbs is called its absorption spectrum. Okay, let's talk about the light reactions. Okay, so the light reactions really start in photosystem 2. Know that they start in photosystem 2. That's really where we're going to begin. In photosystem 2, photons hit the chlorophyll within that photosystem, and basically what happens is they excite electrons and bring them to a higher energy state. If you're in chemistry, you know probably more about what that means, but yeah, those photons energize electrons. And those electrons are carried over into these complexes. Pretty sure you don't need to know the names of those complexes, but those complexes kind of function in the opposite way as the mitochondria, or opposite as in they function in the mitochondria, where they're actually taking those hydrogen ions and pumping them inside the thylakoid. And remember, these photosystems are inside the thylakoids. So they're taking hydrogen ions from the stroma, pumping them inside the thylakoid. And to get more electrons, we're actually breaking down water and taking the electrons from water and releasing oxygen and hydrogen ions into the thylakoid lumen. So no, if you see right here, my cursor is over it. To get electrons, we are...
I don't have an answer for that.
Okay, thanks Siri. So we are breaking down this water, getting its electrons, releasing oxygen and hydrogen ions into the thylakoid lumen. And we're further bringing even more hydrogen using the power of those electrons into the thylakoid lumen. And then eventually those electrons get moved over into photosystem 1. They get re-energized by more photons in photosystem 1. And this time we're using those electron energies to reduce NADP+ to NADPH. And finally we have ATP synthase, which is flipped backwards compared to the mitochondria. And it's actually letting hydrogen flow out of the thylakoid into the stroma, and it's using that gradient energy to phosphorylate ADP and ATP. So know that the whole process of the light-dependent reactions is generating NADPH, oxygen, and ATP. Okay, so now that we have all that ATP and NADPH, what do we do with it? Well, it goes to the Calvin cycle, which takes place in the stroma of the chloroplast. Okay, so this is basically what it looks like. We take atmospheric carbon dioxide, and we start with a molecule called ribulose bisphosphate. And then we take carbon dioxide from the atmosphere, and both ribulose bisphosphate and carbon dioxide react with an enzyme called rubisco, which is the most common enzyme on the planet. And they get fused together in a process called carbon fixation to make 3-PGA. We then use 6 ATPs to turn that into glyceraldehyde 3-phosphate. 6 ATPs and 3 NADPHs to turn that 3-PGA into glyceraldehyde 3-phosphate. So with an initial investment of 3 ribulose bisphosphates and 3 CO2s, we are producing 6 glyceraldehyde 3-phosphates. Now those 6 glyceraldehyde 3-phosphates, one of them goes on to make glucose, and 5 of them go on to get turned back into ribulose bisphosphate. And that completes the cycle. Then we start again with that ribulose bisphosphate, fixate more carbon, and that's how the cycle works. We are taking this ribulose bisphosphate, fixating carbon, using up that ATP and NADPH that we used from the light cycle, generating glyceraldehyde 3-phosphates, 1/6 of which is actually used in the formation of glucose. An important ratio you should probably know is that for 1 molecule of CO2, we are generating 2 glyceraldehyde 3-phosphates. And it requires 6 turns of this cycle to actually make 1 molecule of glucose. That's photosynthesis. Okay, let's go on to cell reproduction. Okay, so let's talk about cell division. Well, a cell's DNA is called its genome. There are 2 types of cells, somatic cells and gametes. Gametes are the sex cells. Pretty much every other type is a somatic cell, and pretty much most cells in humans are diploid cells, which means they have 2 sets of matching chromosomes. The gametes, sex cells, they're haploid. That means they have 1 set of matching chromosomes. And matching sets of chromosomes are called homologous chromosomes. That means they code for the same traits. Essentially, for example, they can both code for hair, but one may say black hair or brown hair or whatever hair, red hair. So know that homologous chromosomes, they're about the same length, and they code for the same traits. The structure of these chromosomes, we have the DNA wrapping around histone proteins, and the histone complex is called a nucleosome. And when we have a chromosome that's been replicated in the S phase of the cell cycle, we call those chromosomes sister chromatids. And that little connection point between sister chromatids is called a centromere. Okay, so the cell cycle has several phases. The majority of the time, the cell is in interphase, where it grows and replicates DNA. The mitotic phase, or M phase, is the actual cell division phase, and it's a very small part of the cell cycle. So the G1 phase is normal growth of the cell, just normal growth and function. S phase is when the DNA is replicated. Those sister chromatids are made during the S phase. The G2 phase, we get some organelles getting replicated. We want to make sure that the DNA synthesis worked properly. That's what happens during G2. And finally, we go to the actual division phase, the M phase, mitotic phase, also called karyokinesis. So, as Dr. Clark said, good acronym, PPMAT, PPMAT, whatever you want to call it. I'll briefly go through what these are. The prophase is when we get those the centrosomes move to the opposite sides of the cell. We start to get a breakdown in the nuclear envelope. Prometaphase, we get the attachment of the microtubules to the kinetochores. In the metaphase, all those chromosomes are lined up around the center of the cell. Anaphase, the sister chromatids are separated. The cohesin protein that holds the chromatids together is broken down, and those sister chromatids can now be called chromosomes. And telophase is when the nuclear envelope reforms, and cytokinesis is when the cells actually separate apart. Some important things to note is in mitosis, sister chromatids get separated, and distinction between plant and animal cells. In animal cells, the cells are separated with a cleavage furrow, and plant cells, a cell plate is what separates the two daughter cells. Let's see if there's anything I missed here. Oh, there's also a G0 phase where cells are not preparing to divide. A lot of cells could actually be in G0 phase for a very long time, or forever in the case of nerve cells or heart cells, for example. Those do not divide. Okay, so there's various ways to regulate this cycle, and we call these checkpoints where the cell cycle is regulated. And there's several of them. There's three main ones you should know: the G1, G2, and M checkpoints. So the G1 checkpoint makes sure that all the resources are available, and that there's no damage to DNA. The G2 checkpoint makes sure that DNA replication occurred properly. That's the primary role of G2, make sure that the chromosomes replicated properly and that they're not damaged. The M checkpoint makes sure that right before the anaphase, where those sister chromatids are separated, that the alignment of the chromosomes is proper and that they're correctly attached to the microtubules. So remember, M phase is in the mitotic, M checkpoint is during the mitotic phase. G2 checkpoint, well, comes right after synthesis, so it checks that synthesis went okay. G1 checks to make sure that we're all good to go in terms of being ready to start replicating that DNA. So there's two groups of proteins that regulate the cell cycle. We have the positive regulators and we have the negative regulators. So the positive regulators are called cyclin-dependent kinases and cyclins. Essentially, these cyclins are generated when it's appropriate for a cell to move past its checkpoint. And these cyclins bind with the cyclin-dependent kinases and allow the cell cycle to proceed. And negative regulators essentially check to make sure that the cell has achieved the benchmarks needed to go past a checkpoint. The ones that you should know are RB, P53, and P21. They work in the G1 checkpoint, and if they detect damaged DNA, P53 can stop the cell from dividing and attempt to repair DNA. If it's not able to repair DNA, it can actually cause the cell to kill itself, because we don't want cells with damaged DNA to replicate. So this is essentially what causes cancer, is when these checkpoints are not adhered to, and cells go berserk and just start dividing like crazy when they're not supposed to. Couple of vocab words you should know. Genes that code for positive cell cycle regulators are called proto-oncogenes, and when those proto-oncogenes become mutated, they are called oncogenes, and the cell becomes cancerous. It starts replicating out of control. This can also happen when these negative cell cycle regulatory proteins, the RB, P53, and P21, they become mutated, or they just stop functioning. And the genes that can code these proteins are called tumor suppressor genes, and if there's something wrong with these genes, these regulators may not function, and they may not be able to stop a cell from basically replicating out of control, or they may not function to stop a cell that has damaged DNA. Okay, last one. Let us talk about meiosis. Okay, so meiosis is the process which we produce these haploid gametes, the sex cells, and we essentially first have to start with a diploid cell, and that diploid cell will divide once, and at that point it has two daughter cells, both of which are haploid, but both of which have the same number of chromosomes as the diploid cell originally started with, and then those two haploid cells divide again, and you'll eventually get a cell with half the number of chromosomes, and yeah, you'll have four total cells at the very end. So just a way to remember this would be you start with a diploid cell. It's got 46 chromosomes. It replicates all of that DNA, so it'll then have 96 chromatids. Then the homologous chromosomes separate into two cells, and then we have two haploid cells. Those separate again, and you'll get your four gametes. So let's go through that process in a little bit more detail. There are two rounds of meiosis, meiosis 1 and meiosis 2. So in meiosis 1, you have the replication of DNA, and after that DNA has been replicated, a process called crossing over happens, in which the homologous chromosomes come together and non-sister chromatids exchange DNA information with each other, and that's called crossing over. So you get this exchange of DNA information. This contributes to genetic diversity. Remember, this is all happening in meiosis 1's prophase. And then all of these chromosomes kind of randomly align at the in metaphase. They kind of randomly align. This again contributes to that genetic diversity. And in anaphase, this is really important, the homologous chromosomes are separated into two separate cells. So homologous chromosomes are separated in meiosis 1, specifically in the anaphase. So in meiosis 2, we don't get that crossing over again. And this time it's just like mitosis, where the sister chromatids are separated. So again, meiosis 1, homologous chromosomes are separated. Meiosis 2, the sister chromatids are separated. Other than that, pretty similar to mitosis in terms of the order of the phases. Okay, last thing to talk about are life cycles. So humans are primarily diploid dominant, which means we spend most of our lives in a diploid state. We only briefly are in a haploid state, and that's when we're in the sperm or egg cell form. So we're diploid organisms, so we're in a diploid dominant life cycle. Haploid dominant life cycle, which basically means the opposite. The majority of the lifespan of the organism is spent in the haploid state and only briefly enters a diploid state. And finally, we have alternation of generations, in which we alternate between a haploid and diploid state. I think in the lecture this was made really overly complex, and I don't think any test question is going to make it too complex. You should just know that the life cycle just essentially details what is the majority of this organism's lifespan in. Is it diploid? Is it haploid most of the time? Is it going back and forth? That's the point of this section is just describing those differences in life cycle. All right, that covers this crash course. Hopefully you found it useful.